So our first talk on forensic DNA analysis is going to be regarding the history of DNA in forensic science, as well as a little bit of a discussion on the structure of DNA. DNA stands for deoxyribonucleic acid, and DNA contains your genetic information. It is all the instructions on making you, you. It's found on chromosomes, which are located in the nucleus of your cells, and you are looking at a picture of Watson and Crick, who were the first people to describe the structure of DNA. Watson and Crick had published a paper in Nature in 1953 describing the structure of the DNA molecule, so they're credited with that discovery since they were first to publish. In 1980, Ray White describes the first polymorphic RFLP marker, and we're going to talk more about what an RFLP marker is and why it's important to us in forensic science in a later lecture. In 1985, Alec Jeffreys isolated DNA markers, and he called them a DNA fingerprint. An individual's DNA fingerprint is similar to their actual fingerprint in that no two people, except for identical twins, will have the same DNA fingerprint, the same DNA pattern. In 1985, Carrie Mullis developed PCR testing, which is a way to amplify samples of DNA so that you can use them for further DNA analysis in forensic science. In 1988, the FBI started using DNA in their cases. In 91, we had our first paper on short tandem repeats, which is another biotechnology analysis method that is used for forensic science. We're going to talk about that again later in our biotech unit. And in 1998, the FBI launched CODIS, which is a database of DNA profiles from felons and other sexually violent predators. In 1962, Watson Crick and Maurice Wilkins were given a Nobel Peace Prize for their work with DNA and discovering the structure of DNA. One person who was not honored in that Nobel Prize was probably one of the most important people, and that was Rosalind Franklin. Rosalind Franklin was a chemist who worked in X-ray crystallography, which is a way to visualize small molecules. And she took X-ray crystallography photos of DNA, and from those, the structure of DNA was able to be determined. However, Watson and Crick got a hold of one of her X-ray crystallography photos without her permission. She did not give that to them willingly, and however was not honored for her work on that. And they were able to deduce from her photo, which is seen down here on the bottom right, that DNA was a double helix, not a triple helix, which is what they had initially hypothesized. She died of cancer before she was able to be honored for her work, and the Nobel Prize is not given posthumously, which is why she's not credited as well for that. Sir Alec Jeffreys is kind of the father of modern forensic DNA analysis, and he came up with this analysis called DNA profiling, and that's using restriction fragment length polymorphisms, which we'll talk more about later. And what he was doing was taking a section of DNA and cutting it with restriction enzymes and then running those through a gel. And the pieces of DNA would separate by size, and he could use those bands to compare crime scene DNA to suspect DNA and looked for matches in those banding patterns. Transferred this to an X-ray so that it could be kept. And so using this technique, he was able in 1985 to confirm the rape confession, or he was asked to confirm the rape confession of a 17-year-old Richard Buckland. He denied raping a young woman, and by comparing the DNA from Richard Buckland and the DNA taken from the victim, he was able to eliminate him as a suspect. The DNA profile did not match, so he was exonerated. Jeffreys also used samples from other suspects to help convict Colin Pitchfork. His DNA did match samples from the victims. Two young women in Narborough, England were raped and murdered, and the police had asked 5,000 local men to submit a DNA sample. And in comparing that, Colin Pitchfork's DNA matched those that was found on the victim. So he was convicted for the rape and murder of those two females. So Alec Jeffreys is credited with the first exoneration or the clearing of a suspect and for the first conviction using forensic DNA evidence. So DNA structurally is a double helix. It has two coiled DNA strands, and it's composed of nucleotides, and a nucleotide has three parts. It has a sugar molecule, and in the case of DNA, it's a deoxyribose sugar, whereas in RNA, it would be a ribose sugar. It has a phosphate group as well as a nitrogen-containing base. For humans, 99.9% of your DNA is the same as every other human's DNA. There's only 0.01% difference. The sides, or what we would call the backbone of DNA, are made up of alternating sugar-phosphate molecules. So here's an image of what we just talked about. We have a phosphate molecule and a sugar molecule and a nitrogenous base. This right here is considered a nucleotide. Notice that there are some base pairing rules. You have adenine always pairing with thymine and cytosine always pairing with guanine. On the backbone here, we can see it goes phosphate, sugar, phosphate, sugar. So we can see that alternating of patterns. And the part that makes up the rung of the ladder of DNA are those nitrogen-containing bases. And those can be different and have different orders coding for different protein products. Those bases are held together in the center by hydrogen bonds. We'll talk later about the importance of hydrogen bonds, but it's a fairly strong yet weak force at the same time. As we just said on the previous slide, the rungs in the middle of that piece of DNA are made up of nitrogen bases. The four bases are adenine, cytosine, guanine, and thymine. Adenine and guanine are called your purines. And the way I remember this is that Ag is the symbol on the periodic table for silver, and silver's pure. And then the other two are just pyrimidines. I don't have anything good for that. Chargaff's rules tell us that bases always pair A with T and G with C. So these two would always pair together, and these two would always pair together. So you always have a double ring structure pairing with a single ring. And what that does is it maintains the width of the DNA. So you don't have 4 rings and then 2 rings, and you would have really wavy looking DNA. So this maintains the same width throughout the DNA molecule. So now you would be expected to answer questions such as, if you had 20% adenine in a sample, how much thymine is present? Well, we know that adenine and thymine should be in equal amounts because they're always paired together. So if we have 20% adenine, we also have 20% thymine. Right, so if you have 20% thymine and 20% adenine, that's a total of 40%. So the other part that's left is 60. You have 60% of a DNA sample that has guanine and cytosine, and because they are equally found in equal amounts, you would have 30% guanine and 30% cytosine. So there are 2 types of DNA. There's nuclear DNA and mitochondrial DNA. Nuclear DNA is found in the nucleus of every cell, except for red blood cells, which do not have a nucleus. You have 46 chromosomes in your cell, in your nucleus. 23 come from mom, 23 come from dad, and each cell only contains 1 nucleus, minus red blood cells. Mitochondrial DNA is found in the cytoplasm because it's found in the mitochondria, which is found in the cytoplasm. You inherit these only from your mom because you wouldn't inherit those organelles from mom's egg cell. Each cell contains hundreds of thousands of mitochondria because those are involved in energy conversion for your cell. And we can find mitochondrial DNA in skeletal remains. So if we don't have good tissue samples left to get nuclear DNA from, then we would go to a mitochondrial DNA to test. The reason that you don't get any mitochondria from your father is because in a sperm cell, you have nuclear DNA present in the head of the sperm, and mitochondrial DNA is present in the tail. Because the tail is involved in locomotion, you're going to need energy and energy conversion for the tail. When the head of the sperm enters the egg, the tail falls off, and therefore the mitochondria does not enter the egg cell. So all those mitochondria you can thank your mom for. When possible, we're going to want to use nuclear DNA to test or to make a DNA profile from, because to analyze mitochondrial DNA, it's more rigorous, it's more time consuming, and it's a lot more costly than doing a nucleic testing of DNA. Mitochondrial DNA is more simple. There's only 37 genes on it, and it's typically in a circle or a loop. So your body DNA, your nuclear DNA, is a long string of DNA, whereas mitochondrial is not. It's very simple. And we would only use this again when nuclear DNA is not available. So genes are portions of DNA that code for specific proteins. If you can imagine a cookbook, and say you want to make a cake out of that cookbook. You're going to flip to the page where the cake recipe is and just make that recipe for a cake. Your DNA works the same way. You have this long length of DNA, and you don't need to make every product that you can from every gene at one time. So you flip through and find the gene you want that makes the specific protein product that you want, and you only make that. DNA is found in all nucleated body cells, so white blood cells, semen, saliva, urine, hair roots, teeth, bone, tissue, but it is most abundant in buccal cells or cheek cells. So whenever we take a sample of DNA, or we want to take a DNA sample from a suspect or a victim, we're typically going to want to do a cheek swab. So we'll rub the inside of a cheek with a sterile cotton swab. As we said before, red blood cells have no nuclei, and therefore they don't have any nuclear DNA. So if we get any DNA from a blood sample, it's coming from the white blood cells. DNA doesn't last forever at a crime scene. There's a lot of factors that can affect DNA left at a crime scene, such as heat. On a hot day, your DNA is not going to be usable after a long time. Sunlight can also damage DNA, as well as moisture, bacteria, and mold. So it's important that you're storing your DNA evidence properly and allowing it to dry out in a paper bag versus putting it straight into a plastic evidence bag. If you were to contaminate it with bacteria or mold, you're running the risk of introducing the bacteria, the mold's DNA, which is going to completely screw up your DNA profile for that case. Not all DNA evidence results in usable DNA profiles, and kind of obvious, but DNA testing doesn't identify when the suspect was at the crime scene or for how long they were there. There's not a set amount, oh, we got so much DNA, that means they were there for an hour. It just doesn't work that way. So we can collect DNA at a crime scene using a bunch of different tools, such as smear slides, using a scalpel to scrape blood off of a wall, tweezers to pick up hair samples that have DNA in them, scissors to cut cloth or any fabric that might have DNA samples on them, sterile cloth squares you would use in the same way that you would use a cotton swab with distilled water, or filter paper with distilled water to rub on a particular stain to pick up DNA evidence from that. UV lights can show, it will react with semen and urine to show where potential sources of DNA would be, so you would know where to do swabs or to take samples from back to the lab. So how do we collect blood? Because blood is one of your best sources of DNA with the white blood cells. If it's on clothing, we would really prefer to take the whole piece of clothing in, because we can get a lot of information about how the blood pattern is or the dispersion. You typically don't want to just leave behind a garment that has potential DNA or blood on it. You can also use a sterile cloth square to take a sample of blood that has DNA off of the piece of evidence so that you can analyze it in the lab. If you have dried blood on furniture, you're going to send the whole object to the lab, but if it's too big to send to the lab, like it's a wall or a tub, you would scrape the blood sample into a sterile dish using a scalpel or some sort of sharp object that allows that dried blood to flake off. DNA typing is a way to take that sample of DNA and convert it into a series of bands that can be compared suspect to crime scene to victim, to distinguish each individual, because each individual's DNA is going to leave a different pattern of bands. And we'll talk more in depth about this during the biotech part. Only 0.1% of DNA differs from one person to the next. So phenotypically, we look very different from one another, but that's only accounting for 0.1% of a difference in our DNA bases. So we use the regions that are different between us to make a DNA profile for an individual. We also have non-coding regions of DNA. About 3% of your DNA actually codes for a protein. So imagine that cookbook again. Imagine it has 100 pages. Only 3 of them will actually make recipes, our recipes that will make food. The other 97 pages just have complete gibberish. It's not usable. You can't make anything from that. That 97% is non-coding, and it tends to be fairly repetitive, repeating similar sequences over and over and over. 50% of the genome has these repetitive sequences interspersed throughout. It's not like you would look at the very beginning of DNA and find all those repetitive sequences, or the very end. It's going to be throughout. So why would we use DNA profiling? We can use it to identify potential suspects. In the Colin Pitchfork case, there were 5,000 samples taken to see if there were any matches between people in that particular town and DNA left on the victim. We can use it to identify crime scenes as well as casualty victims. If you have a mass grave, you would use DNA profiling to identify individuals in that, comparing it to family members' DNA profiles. You can establish paternity and proving family relations. You would use it to match organ donors. You can't just donate a kidney to somebody randomly. You have to have some similar matching points in order to be eligible for a organ donor match. We can identify John or Jane Does. So if we have a person who comes in, we get skeletal remains, we can use the DNA from those skeletal remains and compare it to family members who have missing people to try to bring some closure and identify those people. We also use it to study evolution in ancestry as well as studying inherited disorders.